Well, welcome back. This is our last class meeting. And we have a second round of presentations. And it seems that we have only one group presenting today. And so we will turn things over to Dhanusha, Supriya, and Poorva to present.
So good evening, everyone. Thank you for being present here and giving us another chance to explain our ideas and our work clearly this time. So we have made like a couple of changes after reading the reviews from all of you. And it really helped us to understand where we were going wrong and what we can change in order to make the audience understand it better. So once again, our topic is to find the activity of active EGFR and active IGF1R in activating the ERK pathway in cancer. So I'll go ahead and tell you what are the key elements in the pathway, and then like what EGFR means, what IGFR means, what ERK is in detail. So starting from the basics, usually a cell grows and it has a controlled cell cycle. So which means every cell has to undergo apoptosis, which is the programmed cell death. Hence, there is no accumulation of cells in case of a normal environment or in a normal human being. But in case of cancer, what happens is there's a failure in the cell control due to some genetic changes or the mutations or some external factors or the chemical radiations or anything that or any other reasons. And once all these changes happens, there is a change in the cell control, and then it leads to accumulation of cells. And here I have listed like a few hallmarks of cancers. So hallmark is nothing but like the main characteristic features of a cancer cell, which you can find in any of the cancer cell. So the first one is like it keeps dividing. It has like no limit up to after which it stops dividing. So there's no cell death to stop its division. Also, it surpasses all the growth suppressors. And since many cells are being formed and then they are divided over time, more growth factors are required for the cancer cells to grow without any control. So in such cases, in case of cancer, many growth factors are overexpressed, and EGF and IGF are growth factors that we will be discussing today. So EGF is the epidermal growth factor, and it plays a main role during the embryo formation because that's when a lot of cells and then the tissues and organs are formed. So it plays a main role there. And then in case of adult tissues, it helps in the cell proliferation, growth, and its survival. And when the EGF levels are changed, when this growth factors levels are changed, the growth of the cells are affected. So if the levels go down, then there's less growth in the cells. And when the levels go up, there's more growth. So in case of cancer, you can find the levels of EGF more because it is required for the cells to divide continuously. And the next element would be EGFR. This is a receptor. The previous one is a protein and this is a receptor. And when EGF binds to this receptor, it gets phosphorylated and it gets activated, as you can see in the image. So this can be activated by epidermal growth factor or the transforming growth factor or the beta-cellulin, epiregulin, or the amphiregulin. And once it is activated, it activates many metabolic pathways like ERK, MAPK, and then JAK-STAT pathway, JNK pathway, and everything. Every pathway has its own significance. For example, the JAK-STAT pathway is important in the inflammatory response to an external stimuli. But for our modeling, we have selected the ERK and MAPK pathway, and we wanted to explore what is happening in that part, like how it affects this pathway, because we already had like a reference to which we can go back to and see if you're going in the right direction. So that's the thing. Moving to the next key element, it's going to be it's going to be IGF-1R. It is insulin-like growth factor receptor. And the shape of the receptor mimics the insulin receptor. That's how the name comes. Like that's why it is insulin-like growth factor receptor. And it can be activated by IGF-1 or the IGF-2. And it plays a critical role in transforming events in a normal cell. So in a normal cell, its expression is not observed much, but in case of cancer cells, the IGFR1 levels are overexpressed. The active IGF-1R levels are overexpressed. So when I say active EGFR or when I say active IGF-1R, I am trying to mean that the receptor is active because the ligand has come and bound to it, and now it is functional and it can go ahead and activate the other pathways, like which goes downstream of it. So that's what I'm trying to mean here. And it also acts as an anti-apoptotic agent, so which is the main reason it is important in the cancer cell survival. And then, okay, what else? Next one would be ERK. ERK is extracellular signal regulated kinase, and it is the main element that helps in the activation of many important metabolic pathways downstream. And it activates specific transcription factors that regulate the gene transcription. So transcription is the process by which the DNA is converted to mRNA. And then we all know that mRNA codes for protein. And if there is any changes, or if there's any anything which goes wrong in the coding of proteins, then it affects the function of the protein because the protein folding changes and everything else changes. So an ERK plays a main role in transferring signal from the cell membrane to the nucleus where the gene expression takes place. So this is a very crucial element, and it promotes cancer growth once again by inhibiting apoptosis, by by the induction of DNA repair and arresting the cell cycle. So in this picture, you can see that the yellow highlighted circle is the ERK, and you can see why it's important, because it activates various metabolic pathways which are very essential. So that's why we have chosen to study about ERK and to learn more about it. We were interested because it's very important. And so that's our objective. So to see how the active EGFR and the IGF-1R levels influence the activation of ERK. In case of cancer, the EGF and the IGF are overexpressed, and they bind to EGFR and the IGFR and make it active. So we are just going to see how these active levels influence the activation of ERK. So I'll just give you the overview of how the pathway looks like how the pathway goes. So first we have EGF, and then it binds with EGFR and then it activates EGFR. Similarly, we have IGF, and then that activates IGF-1R, and then the active EGFR goes ahead and activates SOS, which activates RAS, then RAF, then MEK, and then finally the ERK. So the RAF, MEK, and ERK together, this is called as the MAPK pathway, MAP kinase pathway. So this MAP kinase pathway can be activated by a lot of different pathways as well, like a lot of different factors. And that is also an important pathway as its own. And the active IGFR, once again, it can activate SOS and it can go through the same process, or it can activate PI3K, and this PI3K then goes ahead and activates AKT. This AKT is a crucial one because it inhibits the RAF. So when AKT inhibits RAF, it inhibits the MAPK pathway as well. So this acts like a negative regulator, and hence in a normal cell, we cannot see ERK being overexpressed because the cells are under control and it knows how it has to be regulated under the normal under the normal concentration. The active EGFR also activates P3K, and that's how it's being regulated. So this is the overall idea of the pathway, which this is the overall pathway which we are working on. And so this is the first plot that we would like to discuss about. So in order to understand how the ERK is functioning in different levels of EGFR and IGFR, we need to know how ERK works in normal conditions, like in normal cell concentrations. So wild type ERK is nothing but the activity of ERK under normal cell conditions. This has the initial concentration of the active EGFR and IGF-1R. This is what we monitor throughout the model. And then the next graph would be trying to find the relation between the epidermal growth factor and the insulin-like growth factor. So here we are trying to see if the activity of one receptor is inhibiting or promoting the activity of another one. We are trying to find a direct relationship between these two receptors. So this is a user interface which allows you to change the levels of the active EGFR and the active IGF-1R levels. Sorry. Supriya? Yeah. So this is a user interface that allows you to change the levels of EGFR and IGF-1R. Initially, you can see that they work independently in the left-hand side here. And then the ERK active receptors are being activated according to the expression, according to the concentration of EGFR and IGF-1R. So later, when you gradually increase the IGFR levels little beyond the EGFR concentration, what happens is they co-express and the number of active ERK receptors are more. That's what we are trying to find. So initially they work independently, but after a certain concentration, they co-express and they have maximum activity. And in this plot, what we are trying to do is to compare the time response of active ERK under different conditions, like when we have more EGFR expression or more IGF-1R expression, and or when there's a down expression of these two active receptors, or when there's a co-expression of these two receptors. So in case of overexpression, the red curve that you see here, it takes a maximum time to activate the ERK, but at the same time, it sustains for a longer period of time. So when there is more active ERK, it directly means that there is more chances for the cancer cells to proliferate, because there is going to be a lot of growth factors and a lot of metabolic pathways which help the cell to grow. And in case of co-expression and down expression, equal amounts of IGF-1R and EGFR is there, and then the MAPK is being regulated, and then whatever is being expressed is also being suppressed at the same time, and hence we have less active ERK here. And in this one, what we are seeing is the overall analysis of EGFR and IGF-1R pathway on ERK activation. So in the first one, we can see the EGFR has so in this one, we can see the EGFR has a direct connection by in the activation of ERK in a normal cell. But here we see IGFR alone cannot activate ERK, but when it can be, because you don't see any active ERK here, right? So, but when IGFR is being expressed with EGFR, active EGFR, it works parallelly, and then it promotes ERK activation. And in normal cell condition, there's not a lot of IGF-1R in the cell membrane, so you you don't find overexpression of ERK or anything under normal conditions. So that's what we are trying to see in this plot here, how they work independently and how they work when they are together. And now Poorva will be explaining you how the other elements in the pathway affect the ERK's activation. So Poorva, go ahead.
Yeah, thank you. Next slide. Next slide. Can you go ahead for the next slide? I am not. Yeah, thank you. So we are basically understanding here the processes which take place after the activation of EGFR and IGF-1R. So as Dhanusha explained earlier, that after the activation of EGFR and IGF-1R takes place, there is this dimerization formation. So what dimerization does is it causes autophosphorylation of the receptors, and these receptors, they become the docking sites for these adapter proteins. So here in this case, we have SOS as our adapter proteins. So these adapter proteins, these basically, they are the guanine exchange factors. And these guanine exchange factors, they activate RAS. That is the process. How they activate RAS is when these docking sites are filled with adapter proteins. SOS comes in close proximity with RAS. So so RAS, so SOS, it loads RAS with the guanine, as it is guanine exchanging. It loads RAS with GTP, and what it does is RAS further takes it activates the MAPK pathway, and also takes care of the PIK3 pathway. So when this active RAS, it further activates RAF by phosphorylating it. And when this phosphorylated active RAF protein, it further in process dual phosphorylates and activates the MEK, and MEK further in turn dual phosphorylates and activates the ARK. So this is the process. Next slide. So here, when I explain you the pathway, so what indirectly SOS is doing, it's activating ERK pathway. And when we see, in the right diagram that can be seen, the interactive version, when slowly the SOS, the active SOS is increasing in the environment, in the cytoplasm, the ERK activity is slowly increasing. And in the left side, when it's in maximum, the ERK pathway is at its active position, and the SOS can also be seen in the active position. Next slide. So here, when I told that SOS, it loads RAS with GTP, it activates RAS. So RAS, along with IGF-1R, it activates PI3K pathway also. So that's why instant increase in PI3CK active can be seen, but where the other activities cannot be seen to be constantly active, because there is this negative feedback mechanism that is followed after the activation of ERK, which affects EGFR and not because not the IGF-1R, and that is why the activity of PI3KCA is not affected that much. Next slide. Yeah. So AKT and RAF activity. So AKT is the mediator of PI3K pathway. So it has a really important function of inhibiting the RAF activity. When RAF activity is inhibited, it controls basically ERK pathway. So here it can be seen that when its concentration is increasing, even the RAF activity is like coming to zero. So it's basically acting as a negative inhibitor. Thank you. Next slide. So yeah, that was what I was talking about. That after the activation, like minutes of activation of ERK pathway, it has some other targets also in cytoplasm or nucleus, but there is this one, it's also related, one feedback mechanism takes place, that it has the ability to inhibit dephosphorylate the SOS through P90 pathway, and that causes it to form a negative feedback in the pathway. That is why we can see these increases and decreases in other elements activities like RAS, RAF, MEK activities. That's why they can be observed here. And next slide. So yes, in the paper, there is this PP2A gene family mentioned. So it is a constant supply. So its activity is basically involved in cell division and even the cell proliferations. So its activity is not that much in the process, as even if we increase the activity, ERK pathway is still seen to be like working. So the only thing that can be observed here is it slowly decreases it's just decreasing the ERK path flow. That's it. Thank you. I'll the next simulations will be explained by Supriya. Thank you.
Hello, everyone. So thanks, Dhanusha and Poorva, for explaining the background and how each element or how each pathway is helping us to understand how ERK is activated using receptor EGFR and IGF1R. Once we get a clear idea of how the time responses is, we got to understand that in the initial of 5 minutes to 30 minutes, there is a peak activity or peak activation of ERK. So in order to understand what is happening underneath the connection between EGFR and IGF1R, we need to understand how the signaling works between these two receptors. In case of lung cancer, we have read a lot of research papers, and we have understood that in case of lung cancer, non-small cell lung cancer creates a lot of activity in EGFR, since it is a kind of an epidermal carcinoma kind of a thing, that it creates that it proliferates in the lining of the lung. So in order to understand the growth of the lining of the lung cancer, we have to understand how these elements of EGFR and IGF1R is working. So in this plot of analysis, what we are focusing is the signaling between the crosstalk between EGFR and IGF1R. From the plot, this is a plot which we got. This is a plot which we are trying to replicate from the paper, and from the understanding which we got from the paper, we understood that in case of co-expression, as Dhanusha even mentioned previously, in case of co-expression, there is a high crosstalk between EGFR and IGF-1R. This crosstalk we can see in. Can you guys see my cursor? Just let me know if yes. Okay. So the plot 1 and plot 2. We have a high crosstalk between EGFR and IGF-1R. So when I'm sorry. Okay. So in case of the qualitative parameter which we consider to model the crosstalk, to model the reduced model, the value for which the high crosstalk we are seeing, we can see there are two points where ERK is activated, and there is a crosstalk between EGFR and IGF-1R. This can be depicted. This can be understood from plot 1, 2, and 3. But in case of plot 4, the qualitative parameters which we consider for modeling are in adverse conditions. Like when there is an external factor or external disruption in the signaling of EGFR and IGF-1R, we see the crosstalk is not happening, and there is no saddle point, or there is no stability in the model. So in this plot where the cursor is, we see there is no stability. Whereas in case of 1, 2, and 3, we can see in slight modification or slight changes in the concentration of EGFR, there is a crosstalk happening between EGFR and IGF-1R. So this is what we understood from the phase planes. Going forward with our model and analysis, the takeaways which we got is it is important to have a co-expression of EGFR and IGF-1R in order to proliferate the cancer cell proliferation, and the signaling between EGF and IGF-1 is happening maximum during that time. So as no, I did it, Dhanusha. So yeah, so like going into the technicalities of how each element is doing, and how Dhanusha explained what are the key pathways we are including in our model, it is kind of difficult for us, the biological or the non-biological person, to understand how it is happening in the cell lining of the cancer. So this flow diagram will make us understand how it is actually happening. So over here we have like 2 ligands and EGF present in the lining of the cell. So when there is any EGF in the when there is EGF in the environment, and there is a proximity and there is a proximal EGF receptor available to bind, it creates a complex, and which activates the EGFR. Similarly, in case of IGF, the ligand and the receptor binds and create an active complex. So the line which just now showed is the lining, the cell lining, where all this transaction is happening. So next, going forward, we can see there is a epidermal growth factor and activated epidermal growth factor. We have active phosphate attached to it, and we have activated IGF, insulin growth factor as well. So once it is activated, IRS binds to IGF-1R and EGF-1R, then it activates the downstream of the process. So as Dhanusha explained, and as Poorva also explained, that PI3K is one of a downstream element that has been activated in the process of IGF-1R. We can see in PI3K is activated by both EGFR and IGF-1R. Similarly, GRB2 is one of the element which we did not focus, which helps activate SOS, the son of sevenless. It is one of the most important element that gets activated in a process to activate ERK. So here comes the main element SOS, and then the downstream of it is RAS, and yeah, I mean, you get it, like the RAF, RAS, MAPK, the downstream of the process is activated. And this is how the whole process of interaction is happening under the cell lining, in the cell lining of a cancer cell. And it creates a transcription of ligands, receptors, and growth factors. Yeah. This is the whole biological aspect of what our model is. Yeah, I guess, yeah. So these are the main contribution which our team did. It was a roller coaster for us to move from one project to another and implement the mathematical model, and getting to know how biological, converting the biological knowledge into the mathematical model and the model which we built. So yeah, thank you, Poorva, and thank you, Dhanusha, for all the efforts. Yeah, thank you. That's all from our side.
Can you demo the
Yeah, sure. That's what I was going forward to. I'll just open my NanoHub. It's not open. It is taking time. I'll just share my screen for the notebook which we were working on. I hope that's fine.
Oh, it's okay. We'll wait. Show it on NanoHub.
Okay.
Maybe I can ask a question while we're waiting. So I got a little bit confused. You started out by showing a diagram, which was a feed-forward diagram controlling the MAP kinase pathway. And then at the very end, you showed a diagram which had PIP2, PIP3, and a little bit of expression control of EGFR. How are those related to each other?
Yeah, so our model is based on the diagram that we showed first. That's our model. But the last one, we just wanted to show you how overall it works in a cell, like where it works, like the membrane, or like inside the cell, or like outside the cell, like where the receptors are present in the cell. That's where we kept the membrane, and then we showed which are the ones which are activating inside the membrane and the ones activating outside the membrane. It's just for biological aspect, not from the model point of view. That's why we added it towards the end, so that you can just have like an idea of how it works in the cell. That's it.
Okay, great. So the model doesn't do the feedback. The model is for
It does just the feedback with the AKT.
That was all feed forward in the diagram. There isn't anything that comes back from, you say that AKT inhibits RAF and RAS activates RAF. So you have an incoherent feed forward network, like the simplified one we did in class, which is fine. But there's no feedback in the diagram you showed. There's nothing that couples back from the downstream to the upstream in the diagram you started with. So maybe I missed an arrow, or I didn't catch it.
Yeah, it was all feed forward. I'm sorry. Yes.
So I had another question then. So what is turned on at the beginning of the simulation? Right, your ERK level starts at 0 and then it peaks. What is turned on that makes it start? You start with the inactivated EGFR and IGFR, and then they bind to their ligand. And what turns on what initiates the spike in ERK activity?
So that is initiated when the EGFR and the IGFR becomes active. When the EGF and the IGF comes and binds to the receptors, that's when the receptors are activated, and that's when it starts activating the downstream pathways and finally the ERK.
And how is that done in the model? How do you turn on the EGFR and IGFR at the beginning of this in the simulation? How are they turned on?
I guess it's okay. Just a second.
To answer that question, we actually used EGF and IGF as initial values, initial concentration, which is binding to the free receptor that is available in the environment. That is also a constant which we have taken.
So you model explicitly. You start with all of the receptors unbound, and you have a fixed amount of ligand in the environment, or does the ligand used up?
Yeah, that is there is a fixed amount of ligand available in the for this particular model. We used a fixed amount of ligand available in the environment.
So that's a boundary condition. And so then that takes some amount of time. Okay. And so what shuts down the ERK?
So it's
You
basically
see a spike and then it comes down. So what shuts it down?
So basically, it's a continuous process. And when I told you about the feedback mechanism in the ERK is phosphorylating the SOS. It's dephosphorylating the SOS. That is where its control is taking place. That is why we can see the peaks and then downfall in the graph.
So then I'm confused because there is no arrow showing that dephosphorylation in the diagram. And so I think Supriya just said you don't have that there. So I'm trying to understand what is in the model. Or you showed us a nice feed forward diagram. And the feed forward diagram you show then leads to a spike in ERK that shuts down. And now you're telling me that the reason it shuts down is that the ERK is then basically inactivating SOS, and that's the shutdown mechanism. But there is no pathway drawn that way. So maybe you have something where you relate the diagram to the equations that you simulated, so you could show which equations correspond to which component of the diagram. Maybe.
Can you show the Supriya, can you show the diagram, the basic model diagram which you were following?
So in our model, what we did is we created a deactivation or degradation of EGFR. That is where EGFR is over here. We are doing an degradation of EGFR and degradation of IGFR. That is where we are seeing a decrease in activation of ERK after 30 minutes.
Right. So these are first order kinetics. So where is the activation of IGF and IGFR?
This piece is only handling the IGF-1R. We have another model that says the EGF is activated by the free bounded EGF receptor, and then its degradation is done into gamma EGFR. So these 4 lines are the activation and unbounding of EGF and IGF-1R. And over here in this line, we are deactivating EGFR, and respectively over here, we are deactivating IGF-1R.
Right, but I still don't understand what's used up. What turns off? You'd expect, you said that EGF was a boundary species, so it's a constant.
Yeah.
And
Analysis is
in that case, you would
based on the
expect the amount of EGFR to go up and saturate and stay up. So what is the curve of EGFR or activated EGFR and IGFR look like, the time series for those? Because you only showed the time series for ERK. You didn't show any other time series.
We did show the time series for activated EGFR and IGF-1R. I'll go forward and show it to you.
Great.
Yeah. So over here, so when this is activated EGFR and IGF-1R, when we are increasing the activity of EGFR and increasing the activity of IGF-1R, we can see there is an initial amount and then it gradually decreases because of the degradation of activated EGFR and IGF-1R.
So the initial EGFR is the initial amount of active EGFR. Is that what you're saying?
Yes, the complete model which we are building over here is based on the active EGFR and active IGF-1R.
Okay, but
The
so
whole simulation is done on the active EGFR and IGF-1R.
Maybe it's clear to everybody else. Maybe I'm slow here.
So, I will say it is not totally clear to me. Something that may be helpful is having the reaction diagrams. So for me, I just struggle visually with sometimes with the antimony models, right? They're big, and there's a lot of stuff going on. So something that might be helpful is when you show these plots either in the notebook or in the slides, and I know this is a little bit late at the comment, so sorry, but like having the reactions there next to it, because I think that for you all who have been working with the system, and I really appreciate, I can tell you put a lot of effort into explaining biology more clearly. I have a much better idea of like what you're playing with and like what it's grounded in. But for me, just with like how many species there are, it gets harder to keep track of in each plot. So having the arrows by the sides of the time series might help. It's possible that the disconnect that is happening has nothing to do with anything with the structure of the model, but it's a purely communication barrier between us not being able to have all the ODEs in mind. Because even if you show us a really beautiful diagram, it's hard to keep it in mind when we only get to see it for like 2 minutes as we then go on to the slides. So what might be helpful at this point is if we could pull up a snip of that feed forward diagram and literally have it next to the time series. And then you can talk us through maybe what you're saying there. That at least would help me keep track of what's happening. Thank you.
Sure. Let me, is it visible, the split tool?
So I think it's likely what's happening right now is that you're sharing a specific window. If you're trying to do some type of split screen, you just need to share your screen generally, and that should work.
Perfect.
Yes, we can see it now. Yes, yes.
Okay. So our initial background is EGF and IGF are constant, and we have free EGF and IGF receptor present in the model. So it is a constant value. We are not increasing, or we are not letting user modulate it. It's just a constant value. So once the EGF and IGF binds, it activates the EGFR, and the whole process goes forward from here. But once the ERK is activated and we have EGFR available, active EGFR available, it is degrading to form EGFR molecule. The active is been like it's dissociating into its respective forms. There is a receptor and there is the ligand. So it's degrading. There is a degradation in EGFR, active EGFR. That is why we are seeing a decrease in our time response.
So there's a degradation in EGFR, and you're going to have to help me out here. I'm a cognitive scientist by training, not I don't work with chemistry. Okay, so is it degrading in to the chemical acronyms SOS and PI3K, or is it degrading? So I guess what I'm getting at, and I'm wondering if this is part of the confusion. So we have the distinction. So we have state variables whenever we have a dynamic model, right? And that's exactly how many things are changing in our system of equations. Does this network diagram show everything that's changing in your model, or are there other things?
In this network, we did not mention about the degradation of EGF. It is only the activity of the key elements which we are trying to analyze in our from our model. So there is no mention of degradation or dissociation of EGF, active EGFR into their respective molecules.
In the model or in the diagram?
In the diagram.
Okay.
In the model, we have it. Yeah.
So, so Lindsay has a comment which might help clear this up. And I guess my other comment would just be that if you have something degrading, even if it's not transforming into anything in the model, you should draw arrows pointing away from it into nothingness, or sometimes people draw that little null symbol where you have a circle with a line dashed through it. So basically, right, when we draw one of these diagrams, we can have our biological diagram, which you gave a very beautiful one, and then we have our model diagram. And when your model diagram, you want to have an arrow for everything that's influencing the change of the state variables. So what I'm trying to figure out for the confusion here is if there's arrows or chemical names that are missing, and that's part of the reason why. Can
I go back to the flow diagram which I showed? Just give me a moment. I'll just show that.
This one was mainly built to show like the activity between the elements, like how they are related, like whether this activates or like whether it inhibits its activity or something like that. So that was like the main motive of the diagram, like one of the main motives.
So,
so just to be clear, what you're showing now is the biological diagram, right? This is not, this diagram does not represent your model in terms of the reactions and the equations that you use to make it.
this is this is more detailed. We haven't included all of the parameters that are shown in the model. So we have included IGF, we have included EGFR, we have included SOS RAS, this downstream process we have included. From here, once the transcription has been done, there is a degradation. You can see that there is a degradation of EGFR and IGF-1R.
Sure.
So
Okay.
so we haven't mentioned this thing in the network which we created previously, but it's there in our model, in our simulation.
Okay. So what I personally need as someone who just gets overwhelmed by too many nodes and arrows is that I just need, and then I'll stop talking because I may be taking up too much space. But so basically, generally when we're working with networks, we can have, here's our inspiration, and we're not going to deal with the whole thing because that's just too much, right? And then you can have the bits and pieces diagram where we say we're unpacking parts of our model, but there's an in-between step that you need too, which is, this is our entire model. This is everything that we're including. So what I'm hearing is that your model includes fewer details than the whole biological diagram, which is fair. That's reasonable. That's what we do when we model things. But it sounds like it also contains more things than the feed-forward diagram. Like the feed-forward diagram was just a part. So I think some of the questions that Professor Glazier was getting at was that he was picking up on a disconnect between the dynamics you were showing and the feed-forward diagram. So what we as the audience are missing is the actual network diagram that corresponds to everything that you're showing in your model. So I don't know what we're at in terms of time. If you actually want to, I don't know how complicated the model is, if you want to try to like sketch out or fill in the blanks in the feed-forward diagram, but that's where the confusion is.
Yeah, that will be a great thing. I can do that.
Yeah, I think you fundamentally, you've shown us some mathematics, but there has to be a diagram that corresponds exactly to the math you did. If a molecule is decaying, it needs to be in the diagram. If a molecule is phosphorylating something, it needs to be in the diagram. Every arrow in the antimony code needs to be shown in a diagram. Now, it's fine to start with, as I say, I think Claudia was very spot on. It's fine to start with the inspiration, which is your EGF-IGF feed-forward pathways. That's great. And this last picture, I think, is very interesting about some of the feedback. But if you undo the snipping tool for a second, if you move the snipping tool out of the way and just show the picture that you've got underneath it, it's being hidden at the moment. Okay. So it's part of something here. You mentioned, for example, and I don't think your code showed this, that you have the ligand bind to the receptor, and then the back reaction where the ligand and receptor unbind. And I'm not sure that's what you meant to say, because it didn't look like the code did that, but it's what you said happened. And you have an example of this, although half of it, in your example of PIP2 being phosphorylated by PI3 kinase to make PIP3. Now that's missing something, which is that the PI3 kinase is dephosphorylated in that process. So you don't have the output there, right? It's actually PI3 kinase plus PIP2 goes to the dephosphorylated PI3 kinase plus PIP3. Right, because it's a transfer reaction, it's a phosphorylation reaction. And then there's also the dephosphorylation, right? The phosphatase takes it back. And so you have the cycle of activation between PIP2 and PIP3 as a classic example of this. Now, if you have a receptor bind to the ligand to make the complex, to make the active form, and this was Lindsay, I think, had a comment in the chat about this, then you have to ask what happens to it after you have the active form. It could be the ligand could disappear and you could go back to being the inactive form. The receptor ligand complex could be endocytosed and you lose it entirely. it could actually unbind. There could just be an unbinding kinetics where the ligand is released. But each one of those has different mathematics. And so I think it's important to have a diagram like the one Connor said, which is a literal representation of everything that's in the math. And then, as you say, Connor, I think picking out pieces of it to discuss in detail is great. But at the moment, there's no way to connect that initial inspirational diagram with what the math is. So I think that I don't know about other people, but I'm finding it a little bit confusing to understand what's going on.
Yeah, I mean, I will say, Grant, I was very jet lagged, so forgive me if I forget details, but from the last presentation, I will say I'm having an easier time following this presentation, even if it's not quite there yet, than the previous iteration. Like there's a lot of good information here. I think it really is just a matter of closing the gap between those two network designs. The only other comment that I had is that I think you made an edit on your phase portraits for this presentation. And I think that there was a bug in your edited code because I went back and I checked the previous video. Your previous face portraits were correct, but I think that there's a bug on the ones where you changed the names. So if you look at it, and like I said, you had this correct before, so if you revert your code, it will be fine. But look at the flows, right? All the arrows. So do you see where they're going right now? They're going like way off. I don't know if Zoom mirrors me, but they're going off to one of the corners, to infinities, right? But we know that when they intersect, there should be points where it's headed towards the equilibria.
Yeah.
And also we should expect that as those null clients intersect differently, the flows change around them.
Yes.
Right? Because so I think so I checked your previous presentation. So you did it right the first time. The flows changed.
Yeah, I remember. Yeah.
Yeah, but I just wanted to give you a heads up in case you had changed something in your NanoHub application, because I see that you changed the axes to make them more detailed. That your current ones, the flow is not being updated. It looks like chances are you have the right system for your null clients and the wrong system for your flows, and it's just not updating. So I just wanted to give you a heads up about that before I forgot.
Yeah, I did not mark that. Thank you, Connor, for that.
And again, I think it would be good to have a diagram and the equations here, because the pathways that you showed as a feed forward pathway don't have any steady state. The steady state is 0 for what you described. There is no steady state. And so I'm not sure what you're showing a null cline of here, because to get a non-trivial null cline here, you have to have some kind of feedback. Right? The dynamics of the system you showed before goes to 0 always. And so I'm not sure how this picture relates to the thing you're modeling. The one thing I know is that the IGF and EGFR pathways are not symmetrical with each other. And so here you have a symmetrical set of null clines. So the X and the Y axes can be exchanged. It doesn't change the picture. And so I don't know how this relates to the model that you showed. I was a little bit surprised coming back to thank you, Connor, because I was going to ask the same question where the flow doesn't match the null cline. But coming back just a second, when you say that the shutoff of the ERK star is due to the decay of IGFR1 active and EGFR1 active, that surprised me a little bit, because if that's the case, the inhibition inhibition that you showed through Akt isn't doing anything.
Mhm.
In other words, we did the example of an incoherent feed-forward network in class, which is what you have here, right? So that the PI3 kinase turns on Akt, which turns off RAF. SOS turns on RAS, which turns on RAF. So if the SOS RAS pathway is fast and the PI3 kinase Akt pathway is slow, then what you'll get is a pulse, which is what we did in class with the pulse generator. But what you're saying is none of that makes any difference. What's happening is the input signal goes away and everything turns off. And so that depends then on the timescales of the equilibration, which you didn't really discuss. And so I'm trying to understand, when you showed that initial pulse, you say that pulse was entirely due to the disappearance of the upstream factors, and not, in which case I don't know what the Akt pathway is doing at all. So again, I may be missing something. I'm probably missing something here, but I think maybe some more, as Connor said, maybe some more diagrams and showing some of those components independently might make it a little bit clearer.
So this was like a previous version where we had like the old pathway and everything. So we did make a couple of changes after that, but that has not been updated yet. So this is.
Oh, sorry, maybe go back a second, scroll up a little bit.
Yeah.
Where you show the diagram again. So here, Connor, take a look at this, because there's a little bit more detail here. There's still no inactivation of the EGFR and IGFR signals. You have different notation from the normal arrows that we use.
Yeah, so can I ask a question about the arrows, and you can correct me if I'm reading them wrong. So in class, we talked, like, we had the lollipop arrows for activation, right? The ones with the rounded heads, flat-headed ones for inhibition, and then we had arrows, normal arrows for material transfer. So that could be like transforming into something or decaying. In this case, are the black arrows supposed to be like the lollipop arrows, and the red ones are supposed to be like the flat-headed ones?
Well,
Yes.
arrows are representing the activations, and it's the opposite for the dotted arrows.
Okay, so they're not meant to be material transformation. Like, it's not flow of matter in this case. So like, right, so remember, like, we would say, like, in class, like, okay, if X activates X, that was usually a compressed scheme for saying, like, there's a binding and then there's a synthesis of chemical based on that binding, right? So, and usually, that doesn't necessarily involve conservation of mass or anything like that, depending on how we're going about it. Whereas if it's a material transformation, it's like chemicals are actually reacting. So for this diagram, all I'm trying to figure out, as someone who doesn't know where it came from, is how to translate the dashed red arrows and the solid black arrows into the types of arrows that I'm aware of. So, like, for example, you have EGFR goes to SOS. Is that's activation, like the rounded arrows that we looked at in class?
Yeah.
Okay.
Yeah.
Okay. And okay.
Dhanusha, can you go to the equations part where we have it there?
Yeah, maybe what we can do is that if you want to, are those the equations for that diagram?
Yes, yes. These are the equations which we used in the diagram as well as in the model. But we made sure that all the inputs what we understood is there in the it's there in our it's there in our code. The current version we which we are publishing.
Yeah, we are welcome to any other suggestions or questions.
So, I mean, maybe you can just unpack the equations a little bit, and that might clear. So I see on the right, we have kinetics reaction, and then kinetic law, it's mass action versus EMM. Like, I guess I'm just really trying to figure out what the math behind the model is. And the reason why I was looking for the screenshot side by side is that basically what I want to see is that for each thing you have listed in the chemical reactions, I want to be able to look at the diagram and say, ah, yes, there it is. Like, this is where it is upstream. And whatnot. But also, like, and in terms of, like, you know, biology is complicated. It does take practice to get used to translating these things over. But
I mean, yeah, so I guess, I mean, here I guess you've screenshotted this set of equations from a paper.
The one which we are trying to
Right.
replicate.
Right. So
Which
this is
we-
this is the notation we said was confusing.
Mhm.
We spent a couple of classes on how confusing this notation is.
Mhm.
So maybe it would be worth converting it to the standard notation that we use so that it would be easier for somebody to understand.
Okay.
Because what you have here is, if you look at that first equation, Connor, I think, right, you have a single K sitting on the arrow rather than the rate law.
Mhm.
And then it's labeled, I assume EMM means Michaelis-Menten,
Yeah, Michaelis-Menten.
which is not defined though. And that means that there's a saturation constant, which isn't listed.
Mhm.
And you have something which is sort of confusing, which is EGFR active. I guess star always means active here.
Yeah.
Okay. I think Lindsay made that comment that there should be some distinction between the statement that star means the active form always. It's not the standard notation.
Okay.
And then, but you have EGFR, activated EGFR plus SOS goes to activated SOS plus activated EGFR. So the EGFR is on both sides. It's not doing anything. The only reason it's there at all is because it's got to show up in the Michaelis equation somehow. But I still have no idea what the rate law for that would be, given that. I don't know. I wouldn't know how to write the rate law. So I don't know what the law for that. Why don't we look in the map? What is the rate law for activation of SOS? Can you just go to the antimony and let's look at it together for a second.
Yeah.
No, don't unhide. No, leave it up. Leave the code. Leave the screen share up.
Oh.
So where's the antimony code that corresponds to the first line of that table?
Go up.
Can you go to the edit mode? So the first line where we are seeing the SOS conformational activation, It is using the DSOS. It gives rise to the active EGFR. It binds to the active EGFR and it gives to the active SOS that leads to the activation of RAF and EGFR. Yeah, I mean.
And what's the rate equation? Just on the right. It's hard to see
yeah.
because it's long. No, in the first line.
The first line, yes.
Can you scroll to the right so we can see the equation? Scroll to the right.
Scroll to the right.
So you have. So that looks like a Hill function rather than a Michaelis function. Okay, so I think maybe you could explain a little more what the structures of the equations are. I think it would help. Maybe a little bit. Okay. So, and then can you show us where you did those phase portraits? Because you showed the null lines in the phase portraits. Where do you have code that generates that in the app? So why don't you start with the equations? Let's look at the equations today. Go up a little bit. So where did these equations come from? How did you say this is a reduced model? Where does this reduced model come from?
In the paper, the reduced model is what they have described is they are taking the crosstalk happening between elements X and Y, where X is the can you share the name of the paper?
No, it's okay. It only matters what's in the code. Nothing in the paper matters. The only thing that matters is what's in the app. So tell me what's in the app.
Sorry, that was me. That was a comment in chat from me. Just did not mean to derail.
I mean, please, no, please do share the name of the paper. But from the point of view of a person using the app, the only thing they know is what's in the app. So if you show mathematics in the app, it has to be described in the app, not referenced somewhere else. So I'm not sure you have a symmetrical pair of cross activation here, basically, right?
Yeah. So over here, X and Y are the EGFR and IGF-1R that is cross activated, like how Y is IGF-1R is activating EGFR, and how EGFR is activating IGF-1R. So here the A stands for
sorry, hold on a second. EGFR activates IGFR?
I mean, like for the signaling of active EGFR and EGFR and IGF-1R.
Where does that, is that in the diagram? I don't see that. Is that somewhere? Maybe I'm confused.
Signaling we haven't
this?
mentioned in the network.
So this is something else. This isn't from the network you showed.
There's the crosstalk between that is happening. We are trying to understand.
And if you run this, does it generate? Connor, let's check. You notice that the null lines in the phase portrait weren't working. Does it work here in the code? If you run this cell, what happens?
Dhanusha, you just need to run the block of code which we have it there.
I mean, are the libraries imported and good to go?
Yeah,
that you need? Okay.
Oh,
You need to do your load function at the beginning. You could probably run all of them, right? You want to
Yeah.
jump forward to them. I don't think it takes that long to run all of it. Because Connor, you thought maybe there was a bug from the previous talk, so I wanted.
I mean, so yeah, I went ahead and I checked. I think they did it right. I double checked because I wanted to make sure my jet lag wasn't making me misremember. But if I go back to your previous talk, the flows and the null clients look like they correspond, at least at a quick glance. But here they don't, right? And we can tell because, right, two hints. One, whenever the null clients cross, we have a bifurcation, right? Something has emerged that wasn't there before. We get equilibrium points, right? And we see the emergence of equilibrium points, we would expect for there to be a shift in the flow. And here, no matter where you wiggle them, the trajectories that you get look the same. So that corresponds to saying, no matter what I set the parameters to, I get the same time series from the same initial condition. So I think that probably when you, for some reason, those just aren't updating. So, but your null clients are updating. Like the null clients are very visibly changing in the way you described. So that's good. So if you go up to the code, though, we can try to figure out why your flows aren't updating. Okay. So just for us, I think that you programmed this similar to maybe how Professor Glazier programmed his null clients and flows. I do it slightly differently though. So could you just walk me through kind of step by step? I see that you have your mesh grid of the X and Y variables, and then you have U and V, which become the vectors for the rate of change for in each dimension, right? And then you have your model with the equations. So from that point forward, if you could walk through what your code is doing, we can figure out what shifted.
can I share my screen if that's okay? No.
Stay with what you have, please.
Yeah.
It's confusing to switch, so maybe you should let Connor, you walk through with Connor what you think.
So, okay, so you have RP, you store as your antimony model, right?
Yeah.
And then you have something called models. And so
Why don't you just tell us what you got? But let's see first. Just explain to us what the code does.
So we built a model over here, which is taking the equation which we described in the image above, and we are passing the qualitative parameters which we want to check the interaction between EGFR and IGF-1R. So NX and Y are the Hill function of it. T1 and T2 are the threshold for the activity between signal transduction between the two elements which we want to analyze. So model over here, dictionary gives those two inputs. Going forward to the for loop, I'm taking we are taking a mesh grid of 100 points, and we are assigning the values of IDX, and we are pointing the values, and in the value of u, we are putting the derivatives which we got from the model which we created above.
Would you mind showing the model once more? If you scroll. Oh no, or sorry, the anemone code. Oh no, sorry, back where that plot was. Sorry, I should have been more specific than general anemone code, but where you're doing the plotting again, if I could just see, yes, right there, yeah, where it says phase model. All right, never mind. Sorry, I thought I saw. I thought I saw where the error might have been, because I'm wondering why they're just all shooting off like
Yeah,
that. And I thought it was maybe.
Yeah, I got to know the error which we have it in the app, and I was trying to show the same thing, but professor said not to share. So in
where in X and NY set model key. So you set the exponent.
So
And then model key 2 is the initial values of T1 and T2.
what is so on there's no lines, dang it. So if you go right down, so in RP
You can turn on line numbers. Why don't you turn on?
I mean, I think I can guide them to it if, but I don't know where that is. So you'll have to tell them.
I think it's view and then
Okay, perfect. Okay, so clarifying question. You have your keys for the parameters you're passing to the model, and then for lines 33 through 37, you're loading the model with those parameters. Is that the idea?
Yeah.
Okay. So on line 40, you have rp.x. What is rp dot? I see rp.nx and whatnot before, but I don't see what rp. I might just be missing something in your code. I guess I'm trying to figure out if that comes in before. Oh, I'm sorry. That's me forgetting basic antimony stuff for a second. So you're setting the stuff there, and then I'm trying to figure out why the flow is, what's that?
Where is the model run?
So, I mean, it looks like what they're doing is not running the model, but taking the basically just inputting the values into the ODE stored in the model.
So they're just calculating the rates.
Right, but so what I'm wondering is, so you have 4 mesh grids, right?
Yeah.
Which one is supposed to be the values of the state variables, and which one is supposed to be the rate of change for?
The derivatives is stored in u and v, and the values, initial values are x and y.
I mean, if you go down a little more to where you were before you scrolled up, aren't you storing?
Yes.
Okay,
that's
okay,
what
I
the
see.
yeah, I mean, where while putting the values of in the line 41 and 42, if we pass x dash and y dash, will give the desired output which we showed in the last presentation.
Okay, so I
was trying to show earlier.
The general logic of how you're doing this seems sound, which makes sense since you got the correct face portraits the first time. I don't know if anyone else sees. Basically, what's going on is that we have some type of bug that's resulting in them plotting the same flow every time, but different null clients. So maybe let's go to where you're plotting to, and see if it could be something there.
So the null clients are being calculated by hand. They're not coming from the
Model.
model.
Right. So let me ask you a question. If we look at the flow, this might give us a hint. If you look at the flow, do you recognize the flow? Like, is it the flow from the last field that you calculate or something like that? Like if you scroll down to those graphs, I can actually compare it to your old presentation.
Can I share my screen if that's okay?
Okay, go ahead.
Actually, these don't look like any of the flows you had in your last presentation.
Yeah. This was the one which we used.
Wait, this is correct.
Yeah, this is the one we used. And
Okay, so wait, and this is your up-to-date notebook? Okay, so okay, then I apologize. Am I just making you spend time solving a problem that you already solved?
yeah, no worries.
Okay.
So, over here, what we are doing is we calculated the values and we passed the simulated RPX value to the derivative which we have mentioned in the model, sorry, in the antimony, and storing it in U. And that's how we are plotting the stream plot in these plots, this graph.
I'm sorry, I misunderstood completely. I thought you said the opposite. That this is okay. So you were showing the old code, and this is the new code.
Yeah, the plot was
So we did not upload like the new one yet. So we did not publish the new one yet. So whatever opened for me was the old one. That's what we were trying to say. Like I was able to open the old one and we have not published the new one. Like she has the updated notebook, but we still have not like published a new version yet. We are planning to do it by tonight. Like we'll try to modify the diagram. Like we'll try to change the arrows and everything, like the way we depicted it and wherever we were going wrong, misguiding. So we will try to change that and make like a couple of changes and publish the new one.
I'm sorry, I misunderstood, but Supriya, I thought you were saying that what I heard was the opposite of what you said, I guess. Okay, well, that's a lot. You've been very patient to explain this. Are there any more questions from people about this? Okay, well, thank you very much for coming back and giving another round of presentation. And thank you to everybody in the audience for listening. If you have any suggestions there isn't a formal request for comments, but if you have any requests for ideas for improvement or suggestions, I'm sure Supriya and Poorva and Dhanusha would appreciate them, but that's not required at all. And please make sure that I know Lindsay and Grant, you could put it on NanoHub, but if you could put it somewhere that's publicly available, that would be good.
We were able to publish it to NanoHub.
Yeah.
That's
Oh, you
working.
were able to get it too? They fixed it for you?
We scrapped Delirium entirely.
Okay, well, that works too.
Yeah.
I'm sorry to have put you through all of that library compatibility is a bear, huh?
Yeah, and even that, like, just weird things are different between NanoHubs and local versions. Like, even matplotlib had like some differences that would work on my VS Code, but not NanoHub. So watch out, everybody.
I wonder what version of matplotlib they use. I would have thought matplotlib stabilized decades ago, so it would be something where you have old versions.
People always find a need to update things, even if the functionality ends up being roughly the same.
Well, I know the IPy widgets are strange on NanoHub. That one we've done. But I didn't realize there was a matplotlib defect on NanoHub too.
It was just a very small, like, notational thing. Like, one of the keywords that my matplotlib accepted was not accepted by NanoHub's matplotlib.
All right. I'm sorry you had to go through that. I understand everyone that making these things publicly available is a chore, but it is important. So then, thank you, everybody. I appreciate all of the patience. Please try to get the final versions of everything up for tomorrow. If you've already got it up, that's great. Let us know. But if you need till tomorrow early afternoon, we can wait till then. If there are any final tweaks that you want to make. Otherwise, I want to thank everybody, and I wish you a restful holiday. And I'll see some of you next year, I know. Appreciate it. And I'm meeting with Peter after class. So I'd be happy to, I'll stick around, I'll turn off everything and meet with Peter.
Thank you, Professor.